Britannica defines synthetic biology as a field of research in which the main objective is to create fully operational biological systems from the smallest constituent parts possible, including DNA, proteins, and other organic molecules. To make it simpler, synthetic biology aims to create living systems that can resolve current issues on this planet by assembling living organisms such as bacteria and fungi into a system analogous to an electrical circuit, so it is the integration of many technologies. The history of synthetic biology can be traced back to the year 1828 when the German chemist Friedrich Wöhler applied ammonium chloride to silver isocyanate to produce urea. This was the first time in history in which an organic substance is synthesized from inorganic compounds. It wasn't until 1978 that recombinant DNA technology was introduced that totally changed the way biological engineering operates. Implications of synthetic biology range from insulin to biodegradable plastics. Insulin was first purified by Frederick G. Banting and Charles H. Best in 1921, before the revolution in biotechnology in the 1980s. Scientists extracted insulin from cows or pigs now that they use bacteria to produce this protein. The primary ingredient found in biodegradable plastics made up of corn is polylactide, and it is made by microbes from cornstarch. There is a hierarchy in the system of synthetic biology. Synthetic biologists use parts as their building blocks, and it refers to a sequence of DNA that encodes for a specific biological function. A device is composed of different parts, and ultimately devices will be fitted into a system. Molecular tools are important in synthetic biology. They are used for reordering and creating new sequences. For example, CRISPR, which stands for Clustered Regularly Interspaced Palindromic Repeats, has become the most popular and cost-effective gene editing tool. It is composed of two parts, sgRNA and Cas9 protein. sgRNA guides and Cas9 proteins do the cut. Please check out our next video for details. Model system is extensively studied and used in scientific research, and hopefully, the discovery from model organisms will provide insight for the work on other organisms. E. coli is particularly common in molecular experiments. One reason for that is because of its fast reproduction speed as a bacterium. E. coli can be used to produce proteins, as long as you insert the right DNA, which is the commander in the protein synthesizing factory, which is the ribosome. DNA sequence data is important for comparison and analysis. Sharing database allows researchers to use the existing information for innovations, as there is a famous saying, standing on the shoulders of giants. Some of you may have realized that it is pretty similar to genetic engineering. Actually, you're right. Synthetic biology and genetic engineering do share an essential toolkit, but the major difference is the scale at which changes are made. Synthetic biologists redesign natural systems to achieve their goals, for example, producing a drug by taking advantage of fast-growing bacteria, and sometimes even use the metabolites of bacteria for diagnostic reasons. Genetic engineering tends to investigate specific genes and how they would affect the system if they were knocked out or inserted. So that's it for this video. Please like and subscribe and check out our next video on the history, origin, and science behind CRISPR, a synthetic biology tool.